Good evening, everyone. Am I audible? Please confirm if I'm audible, students. I hope my PPT is also visible to you. So, today's topic is Unit 5C, and the topic name is Organogenesis. This is from Unit 5, Developmental Biology. We'll be discussing the CSIR NET question answers from Part C. Okay, let's begin. The first question is from Drosophila development. It says Antennapedia complex in Drosophila. It has 5 genes. Antennapedia complex, they are a set of homeotic genes, if you would know. Homeotic genes, they control the organ development, and they are conserved regions of genes which are present in a stretch or in a series. Okay, so these 5 genes, they are part of a complex: labial, proboscipedia, deformed, sexcom reduced, and antennapedia, and they are expressed in parasegments 1 to 5 in a non-overlapping manner. So non-overlapping means on the sides. Okay, so in the larva or in the later stages of development, the region of antennapedia expression, it corresponds to the part of second thoracic segment. Okay, so mutation in antennapedia, it is known to cause the transformation of antenna to leg-like structure. So what does that mean? If there is a mutation in the antennapedia, generally it will control the development of the thoracic region of the Drosophila, where legs are supposed to come out, okay, and in the mouth part antenna comes out. But if a mutation happens in antennapedia, then from thoracic segment legs will come out, but also from the mouth region antenna will also come out. From the mouth region leg will also come out instead of antenna. So with below are certain statements made in respect to the functions of antennapedia. So in this type of described mutation, the gene is ectopically expressed in the head region. Ectopically means where it is not supposed to be present, but still it is expressed over there. That is what is ectopically. Okay. And one of the functions of antennapedia is to repress the gene that induce antenna development. So you will have to tell which one is correct. Okay, and also this option that they have given, antennapedia expresses in thorax and forms a concentration gradient in the posterior anterior direction, affecting head development. A homozygous recessive mutation of antennapedia is expected to transform the leg to antenna in second thoracic segment. So out of these combinations, which is the right one? Yes, anyone? So clearly we can see that whatever antennapedia was supposed to do, it is affecting the mouth part also, and mouth is in the head region. So A statement has to be correct, isn't it? That the gene ectopically expresses where it was not supposed to be present, but still it has expressed over there. This must be a gain of mutation, gain of function mutation. Gain of function means what? That it will be overexpressed. Apart from its regular region, it will be overexpressed in that area. Okay. Then B is one of the function of antennapedia is to repress genes that induce antenna development. Even this is correct because it was supposed to make antenna, but instead of that, it suppressed it and made the leg-like structures. So this also has to be correct. A and B is correct. Now, if A and B is correct, you have to find either C is correct or D is correct. Okay. So antennapedia expresses in thorax. This part is correct. And it forms a concentration gradient in the posterior anterior direction. See, if it was normally, it is forming posterior anterior direction. If it forms gradient, then don't you think that the leg-like structure in the Drosophila, it will be formed everywhere from thorax to head. Correct? In the thorax, it was expressed, naturally, it is expressed normally in wild type, where it produces leg-like structure. Now, if there is a overexpression and it is in a concentration gradient, then maximum expression of ectopic legs would have been before the head, in between the head and thorax. But here the expression is maximum in the head region only. That means this gradient part is not correct. It doesn't form a concentration gradient. Okay? It does affect the head development, but in a different manner, not in this posterior anterior direction. Alright? Now, homozygous recessive mutation is expected to transform. So, the homozygous recessive means a loss of function. Antennapedia was supposed to be ectopically expressed in this particular mutation. It is ectopically expressed in the head. But what if it is not there? If it is completely mutated, then it leads to homozygous recessive mutation. So that is expected to transform the leg to antenna. In this case, the leg will be converted into antenna, because in the thorax also, you will not have antennapedia. So leg also will be converted to antenna in the second thoracic segment. That's why D is also correct. Here A, B and D is the correct answer. Okay. So let's have a look at the theory part of this question. Homeotic gene, they control the development of segmental organization of the body structure. If this is the Drosophila embryo and some homeotic genes are expressed in this sequence, in this exact sequence, you will also find that in the adult fly structure, in the anterior part, wherever this gene was expressed, its product will be head-like structure. Wherever this is expressed, its product will be thorax-like structure. And wherever this is expressed, its product will be involved in expressing the abdomen region. Okay. So these are all the homeotic selector genes. These are nothing but transcription factors which can help in the gene expression of the required molecules of that region. So for example, Antennapedia is involved in making the thorax region. So Antennapedia as a transcription factor will be activating the gene expression that is ultimately going to form the thorax-like structures. Okay. Now, the homeotic gene complexes in Drosophila are divided into two sets. One is this ANTC or Antennapedia complex, in which these 5 genes are present, and the sequence in which they are present in the gene, in the same sequence they will be expressed in the body as well. For example, labial will be in the mouth region, then proboscipedia in this oral part, deformed area in the entire head region, right? And sex chrome reduced in the first thoracic segment, Antennapedia in the second thoracic segment, ultrabithorax in the third thoracic segment, and ABDA in the abdomen, and ABDB also in the abdomen. These all is part of the another complex, which is bithorax complex. Okay, so that's the characteristic of this homeotic genes. Fine. And this is a very good diagram to understand what exactly homeotic gene is doing. Let's say in the adult fly, this particular segment, which is the A6 segment, it is determined in by this gene present in each of the cells of this Drosophila. Okay, in this entire Drosophila body, adult body, how many ever cells are there, all will have this A6 gene. But this A6 gene will not be expressed in all the cells. Gene will be there, but it will not be expressed. Only in this area, that is A6, A7 and A8, these gene will be activated. They will be expressed. And it's because of the presence of this gene only that these segments are being able to be made. Okay. The molecules will have some kind of physical feature here. They are making some physical feature, right? So that physical feature is possible because of this transcription factor, which was originally expressed only in this region of the larva when the Drosophila was in the embryonic form. All right. So as I mentioned to you, this is a selector gene, Antennapedia. It is normally turned on, which in genetic terms means expressed in the thorax, and turned off, that is repressed in the head cells, normally in the wild type. But if there is a mutation, however, mutations in Antennapedia can cause it to turn on in the head, and form a pair of legs where the antenna would normally be. All right. So this is what is the antenna. Okay. You can see how it has been transformed into legs. See, legs are coming out from the antenna region. The antenna is not there anymore. All right. So gain of function mutations are generally dominant. They can result from overexpression of a gene or ectopic expression, which is expression in the wrong place. Another type of gain of function is the dominant negative, which is the product of the mutant gene that can compete or inhibit the function of a wild type product. Okay. So this gene was identified for the mutant phenotype. Antenna means antenna. Pedia means leg. Because the antenna is converted into leg, that's why this gene is named like this. Mostly in Unit 5, it's like that only. Development biology, whatever gene names are there, they are mostly named by the mutational phenotype that they cause. So this gene is not required for antenna formation. Don't take that it is Antennapedia because it is required for antenna formation. No, it's the other way around. It turns out that this is dominant gain of function which causes ectopic expression of the Antennapedia gene in the head. Normally, it is most highly expressed in the second thoracic segment where legs form and are not expressed in the head at all. So, recessive loss of function alleles of Antennapedia give the opposite phenotype. That means antenna will form in place of legs on the second thoracic segment. Okay. Now, next question is based on again Drosophila development, organogenesis. Wild type Drosophila, even this is regarding homeotic gene only. So wild type Drosophila, they have a pair of wings of one segment and a pair of halteres on the adjacent posterior segment. So generally what happens for homeotic genes, each region they have their own set of molecules which they will express. So for example, if I show you in this, see this is T1 or thoracic region 1, this is T2 or second thoracic segment, and this is T3 which is third thoracic segment. You can see from each of the segments one pair of legs have come out. This is one pair, this is one pair, and this is one pair. Okay. But the wings, they have only come out from the second thoracic segment. See, second thoracic segment, they have a pair of wings. And third thoracic segment, they have a structure which is called as haltere. It helps them in the maintenance of balance during the flight duration. Okay. Now, the homeotic genes, specific homeotic genes are expressed in specific regions of the body. Like, let's say this is the Antennapedia. Okay. Can you see Antennapedia is expressed only in the second thoracic segment? You don't see it expressed this side or this side. Means here it is expressed, but along with that some other genes is expressed. But exclusively only Antennapedia, it is only in the T2. That is why only from the T2 these wings are coming out. Okay. Similarly, Ultrabithorax, it is exclusively expressed in this T3 and A1. Along with it, Antennapedia is also there. So together they are producing a kind of combination. But whatever it is, they are controlling this haltere production. So that gene which is it is controlling the haltere production. You can see this green color. It is the UBX gene, which is called as ultrabithorax. So let's say this UBX gene is mutated. Then in that case, what will happen? Haltere production will be affected, right? As I said, Antennapedia is present here also, along with T3 and along with UBX. Antennapedia is expressed in T3 and A1 also. But UBX presence in this region along with Antennapedia is what makes the difference. But if the ultrabithorax is not there, then it's just a repetition of the Antennapedia. That is why here also pair of wings will come out. You will have 4 pair of wings. So that's how the homeotic genes, if they are misplaced or they are misexpressed in some areas where they are not supposed to be expressed, then such kind of mutational effects are generally seen. So this question is about the wild type Drosophila. It's from June 2024, last exam itself. Okay, they have a pair of wings on one segment and a pair of halteres on the adjacent posterior segment. Adjacent posterior means they have not mentioned over here. So this is in T2 segment, this is in T3 segment. Okay, now you know. Wild type 4-winged insects like dragonflies, they do not have halteres. So you would notice that if you take Drosophila, the small fruit fly, it has only one set of wings, one pair of wings. But dragonflies, they have 4 wings. You know, right? 4 wings they will have. So why they have 4 wings? The reason is because they don't, that's what is based on this concept, okay? So wild type dragonflies, they don't have haltere. Instead, they have these wings. So UBX is a homeobox gene. UBX mutants of Drosophila have 2 pairs of wings and no halteres. In fact, if you take UBX mutants also, ultrabithorax, for the same reason what I mentioned to you, for them also, If the UBX mutant is there, then you will develop two pair of wings in Drosophila as well. So in relation to UBX function, the two-winged and the four-winged insect species differ based on what? Okay, what is the reason that their function differs? Is there UBX expression levels in segments? The different expression level that give rise to wing halt or haltier, or it is the regulation at different developmental times that sometimes they will give wings and sometimes they will give haltier, or is it the target of the UBX in segments that give rise to wings and haltiers? That means where the UBX will be expressed, or is it copy number or paralog evolution? Anyone wants to take a guess at this question? Now you know many things about homeobox mutants. No one? Okay. So this answer is obviously option 3, because as I mentioned to you, the UBX will not be expressed in all regions of the body, only in the regions where different combinations of genes are activated before the homeotic genes. So that would be gap genes, pair rule genes, segment polarity genes. With all these zygotic gene combinations, the targets of UBX will be expressed in different segments. Okay. UBX, first of all, will not be present in all segments. It will be only in the specific segment. And wherever this UBX is present, what this UBX will be controlling, accordingly, the phenotype will be given. So if it is expressed in the T2, if it is not there, UBX is not there, then T2 makes only wings. If it is present in T3, then it will make haltier. Okay, UBX. UBX it is present. But if UBX is not there in T3, then instead of haltier, even in T3 also wings will be made. So that's why this statement is the correct statement. In Drosophila, UBX is expressed at a certain level in the third thoracic segment to repress the wing formation and promote haltier development. However, the same function is not seen in four winged insects like dragonflies, where the third thoracic segment also forms wings. So the difference is not the amount of UBX, but rather how the presence of UBX leads to different outcomes, which relates more to the targets of UBX rather than the expression level. So that is why option 1 can be ruled out, where it says that it is in different, means it will be, yeah. So the difference, the option 1 says that UBX levels will give rise to expression level. It's not really the expression level. It's about what it will target. Now, while timing of the gene expression can be crucial, in some cases in development, timing also produces different phenotype. But the difference between the two-wing and the four-winged is not due to UBX. Okay, but how the UBX influences the segment identity. So that's why timing is also cannot be an answer. Even the copy number or paralog evolution, they can theoretically influence segment polarity if segment identity if the copies had different function. But in this case, the difference between two-winged and four-winged segments is more about UBX functioning within the context of the segments where it is expressed. So option 3 is also not the answer. Option 4 is also not the answer. Option 3 is the most likely answer because the fundamental difference between them lies in how UBX controls the expression of genes downstream of it. What genes will be controlled further, which determine whether a segment forms wings or haltiers. Differences in these downstream targets between species could explain why the Drosophila has haltiers, okay, and why these dragonflies have additional wings. So you can see here the wild type Drosophila, it has only one pair of wings. But if it is mutated Ultrabithorax, it has four wings, similar to what dragonflies also have. Okay. So I just want to tell you about the Lakshya batch. So this is for CSIR NET 2025 and GATE 2026 exam, where you will be getting a complete package of unlimited classes, your doubt support, chat support 24/7. You can get your doubts clarified while you study. A dedicated study portal where all the recordings, video recordings, and PPT will be given, and Conceptica series free access. Even the test series also will be there. All right. Then we have the T3 series going on. It's the test series that Biotecnika is giving to its students. All you have to do is you just have to download the app, Biotecnika Campus app, and there you will be getting this free series. This is free test series. You can avail this because if you are opting for this 1st March exam, CSIR exam, then this will be very beneficial to you. The more and more you practice questions, the more you will be confident when you're attending the exam. Okay, and this consists exclusively only Part C questions, difficult Part C questions to test your knowledge. Right? All right, the next question is regarding mammals. We already saw regarding Drosophila. Following statements were given regarding the decisions taken during the development of mammalian embryos. So, you might be knowing in mammals, you have this ICM, which is inner cell mass, a group of cells in the blastocyst of mammals. You would be aware that once the embryo is formed, embryo undergoes cleavage division from morula to blastula. So, mammalian blastocyst has a group of cells in the center part, which is the inner cell mass or ICM. ICM gives rise to the proper embryo, okay? Whereas the outer layer of cells, which is called as a trophoblast, it generally gives rise to the placenta or extra embryonic membrane. Now, there are some particular factors, transcription factors, which are expressed in ICM and the trophoblast. This outside layer is called as trophoblast. So, they are expressed differently. Okay, these are expressed differently in these cells. So, you have to tell which of these options are correct, which is expressed where, and how they are affecting the formation of ICM. Which of the statements are true among this? Yes. Any guesses, anyone? All right. So I'll explain this one. See, this diagram shows the mammalian embryo, early stages, in the early stages. Outside fertilization envelope is there. The zona pellucida will harden and form the fertilization envelope. Inside few cells you can see, and the nucleus. So nucleus is given some colors, all right, and some cells are present. There are 4 transcription factors which you are supposed to know: OCT4, CDX2, NANOG, and GATA6. Okay, initially, okay, Sarita says option 1. Let's have a look. What is the correct answer? So we have here in the cells initially only OCT4 is expressed, and that too it is expressed in all the cells. I mean, OCT4, what is the maternal? It is coming from maternal side also and zygotic side also. It is expressed in all the cells. Okay. Later, also you can see OCT4 is again kind of present in all cells, but at a later stage. Now this is the blastocyst. We have ICM and we have the trophoblast. You can see that OCT4 is slowly it is inhibited. Okay, where OCT4 is inhibited? In the trophoblast, outer layer of cells that are supposed to form the placenta. So clearly we can understand that CDX2, it must be expressed in the trophoblast, and CDX2 must inhibit OCT4. Whenever we write sign like this means it is inhibiting. Okay. So CDX2 must be inhibiting OCT4. That is why it cannot be expressed in the outer layer of cells. Okay. So that is why we'll have a look first. Let's see what all other things are there. Now CDX2, from initial stages, it has stochastic expression. Stochastic expression means random. It is not expressed in all the cells. You can see few of the cells it will be expressed, but not in all. Okay, but slowly, this will have a connection with the apical domain. Apical domain means towards the surface. Okay, wherever this surface areas are there, apex side, they will promote the development or building up accumulation of CDX2 in those cells. But whichever cells are inside environment, they are towards the basal side, they will not be able to develop CDX2. They inhibit the development of CDX2 or accumulation of CDX2. So automatically, slowly you will see that CDX2 starts getting restricted only to the outer layer of cells. And when it comes to the blastocyst, you can clearly see CDX2 is completely towards the outside, only in the cells of the trophoblast, but nothing is there in the inner cell mass. Inner cell mass doesn't have CDX2 at all. But where CDX2 is present, they are not allowing OCT4 to build up. So since the inner cell mass doesn't have CDX2, OCT4 will be built up over here. All right. Then coming to nanog. Nanog will be present again stochastically, even GATA6 also. Okay. And even later stages also you can see both are building up. But there is a problem with nanog, same as OCT4. It cannot be present in those cells which express CDX2. So you can see that in the outer layer of cells, trophoblast, there is no nanog at all. Okay. But one more molecule is there which will inhibit nanog, which is the GRB2. Okay, now GRB2 will not be there in all the cells. So whichever cells GRB2 will be there, they will promote the development of GATA6 but inhibit the promotion of nanog. That is why, see in these blue color cells, dark blue, navy blue, all these cells have the GRB2 expression, but they don't have nanog because GRB2 inhibits nanog. But they will have GATA6. So these navy blue color cells are the ones in inner cell mass which are expressing GATA6 molecule. But the other cells, okay, so these other cells you can see they don't have GRB2, they are able to express the nanog. So this nanog and GATA6, they are like a salt and pepper expression. Whichever cells have GATA6, they will not have nanog. Whichever cells have nanog, they will not have GATA6. And anyways, outer cell, neither nanog nor GATA6 is present. Okay. So answer is option 1. Yes, Sarita, you're right. It is saying the pluripotency of the inner cell mass. Pluripotency, we know inner cell mass gives rise to the embryonic cells, all the embryonic cells. So the layers that you have, germ layers, ectoderm, mesoderm, endoderm, they all come from the inner cell mass. And they are maintained by a core of 3 transcription factors. OCT4, SOX2, nanog, these are all transcription factors. So they will be expressed. And prior to blastocyst formation, each blastomere expresses both CDX2 and the OCT4 transcription factors. So even this is correct because before even blastocyst formation, I said OCT4 is a maternal transcription factor also, CDX2. So every blastomere, all cells will express CDX2 also and OCT4 also, and they will be able to. It appears to be capable of making either ICM or trophoblast. But some cells have this apical domain thing. They can be only present in the cells where apical domain markers will be there. So that's why those cells become trophoblasts and they start expressing CDX2. So they stop the expression of OCT4. Okay. Both ICM and trophoblast cells, they synthesize transcription factors. CDX2, no, not at all. ICM doesn't express CDX2. Only trophoblast cells express. And OCT4 activates CDX2 expression, not at all. If that would have been the case, OCT4 was in ICM. In ICM also, you would have seen CDX2. But you don't see CDX2 in ICM. Okay? So, yeah. C and D is not correct. A and B, option 1 is the right answer. All right? So, moving on to the next one. In fact, there is one more molecule which is called as STAT3. All right. ICM, they consist of embryonic stem cells, as you know. STAT3 is another stemness kind of factor, you can say. It gives the ICM its self-renewal property. We know that stem cells contain the self-renewal property. Activated form of STAT3, which is generally present in the blastocyst, it stimulates the self-renewal of ICM blastomeres. Okay. Later, nanog prevents ICM blastomere self-renewal in the epiblast and stimulates blastomere self-renewal in the epiblast also. Okay. Next question. So now this is from C. elegans. You all know vulva development in C. elegans is a very important topic when it comes to organogenesis in Unit 5. The anchor cell. So here we have this nematode vulva formation, and it is entire vulva consists of only 22 cells, which actually starts with 6 cells. Okay, so we have 6 cells which are called as the vulval precursor cell or VPC, and later changes in them is what makes this 22 cells. It's a complete signaling process that is involved. There is something which is called as an anchor cell. Anchor cell gives the signaling to those 6 VPC, but only 3 out of those 6 VPC, they start developing to form the vulva. Then why 6 are present? It's pair. It's like a spare cell. If this 3 doesn't work for some reason, then the other 3 will take up its place. Okay. The anchor cell produces lin3 protein during the vulva development, which interacts with the let23 protein present on the 6 VPC that form an equivalence group. Okay, so these are all equivalents. Why are they called equivalents? Because any of them can produce the vulva. It doesn't produce all of them, but at least they have the capacity to produce. Okay. Now the central lineage cell, which is the P6P, actually those VPCs are named from P3P to P8P. Okay. So we have P3P, P4P, P5P, P6P, P7P and P8P. These are the cell names, 6 vulval precursor cells. So anchor cells send signal to all of them, but only the central 3 cells is what forms the vulva. Okay. So the central lineage cell, that is P6P, where anchor cell is closest to, it takes the primary fate, and the cells which are near to that, that is P5P and P7P, they take the secondary fate. Okay. Now few mutants and the phenotypes are given. Can you tell me which is correct? Okay, Sarita says option 1 for this also. All right. So let's have a look. First of all, you should know the anchor cell is sending signal. What is the signal type? That should be clear to you. So the signal name is what is lin3. The signal name is lin3, and the cells which it is giving signal to, that is the P5P, they will have this receptor which is called as let23. Let23 is the receptor. It is an RTK type of signaling receptor tyrosine kinase, which further will activate some of the signaling responses in P5P, because of which the P5P goes for primary fate. Okay. Now the first question is loss of, let's say B, loss of function of lin3. You know it's because of the signaling only that the P5P gets the primary fate. Now if we don't have the lin3, signal itself is not there, but some reason somehow they have gained the let23 mutation. Okay, so this is only loss of function of lin3 A, and B is loss of function, but the signal receptor is activated. Without the signal, that is what is gain of function. So even if the loss of function of signal is there, if receptor is activated, then you will be getting the normal expression. In fact, more expression is possible, isn't it? But if there is loss of function of mutation, then uh, neither the primary fate will be formed and nor the secondary fate will be formed. So in that case, all the VPCs, they will be taking the tertiary fate. Okay, if that is the case, all of them will be taking the tertiary fate. Now, what if there is a reduced function of lin3? Loss of function means neither primary fate nor secondary fate. So actually, anchor cell, when it gives the signal, out of that, the first centrally placed 3 cells, one is primary fate, another is secondary fate, another is secondary fate. The other ones, they will take the tertiary fate. Tertiary fate means they will just form the skin structure, hypodermis. Okay. So if you have reduced function means what? Instead of giving a longer range of signal, the signal will be less. From anchor cell, the signal will be less. Okay. So what can you expect from here? That the P6P, it is getting normal signaling only, because from anchor cell, primary cell is at least closer, no? So it will get the primary fate, but the other ones will not be able to get signal at all, isn't it? So secondary fate also will not be able to be formed. That is why reduced function of lin3 means P6P adopt primary fate and the rest of the VPCs will be getting tertiary fate. Now, what if the loss of function of lin3, that means all VPCs were taking tertiary fate initially, but along with it, if you have gain of function of let23, then even if the signal is not there, but all the receptors in primary cell also, in secondary cell also, all the receptors are activated. That means all are taking the primary fate. In that case, it will lead to multivulva. More than one vulva will be developed. Okay. And if we have a overexpression of lin3, overexpression means the P5P, P6P, P7P, initial 3 ones, So all of them will take the primary fate because this anchor cell is signal is more over signaling is there. So secondary fate will not be formed. All of them receive high concentration of signal, so all of them become primary fate. Whereas the nearby cells, in this case the P8P and the P4P, which were originally supposed to form the tertiary fate, now they start to take the secondary fate. So P4P and P8P will take the secondary fate. That is why D will be option 1. Yes, Sarita, very good. Correct option is option 1. So this is the vulva development in C. elegans. It's an important topic. Here is the anchor cell, the signaling cell, and this is the group of cells VPC, vulval precursor cells, which are going to make the 22 cell vulval structure. See, this is the 22 cell vulval structure. Okay. So anchor cell is closest to the P6P. That is why P6P takes the primary fate. And the nearby cells which get lesser concentration of the signal, they will take up secondary fate. And the cells which don't receive any signal at all, they will take up the tertiary fate. Okay. But, and you can see anchor cell how it is releasing the lin3 signal. Only P6P is getting the signal. And further, why these cells are getting secondary fate? Why are they not getting primary fate even though they receive some of the signal from anchor cell? The reason is because first of all, it's about the timing. The P6P will be the first one to receive the signal, and immediately it will activate the lin12 in the nearby cell. So this is an inhibitory signal. Okay, inhibitory signal to do what? Not to form the primary fate. So once this cell has become primary fate, it doesn't allow the nearby cells to take a primary fate. That is why they go for secondary fate. So there are some differences in the structure, the cells which form primary and the ones which form secondary. You can yourself see. Okay, the structural difference is there. So initially, this entire signaling is one inducing signal from anchor cell to P6P, then an inhibitory lateral signal. Lateral means towards the side. This is an example of sequential induction as well. Only once the P6P has been fate is determined, then only it will give inhibitory signal to the nearby cells not to form the primary structure. That is why they will form secondary structure. This is this is achieved by the expression or the activation of lin12 in these cells. See, vulva vul on lin12 is on, and this is a juxtaprime signaling. It is it is notch delta type of signaling. Okay. So this is how the entire thing is produced. Lin3 signaling via LED23 promotes the primary fate, which induces the production of DSL ligands. DSL is this delta signals and downregulation of the receptor. Signal is name is DSL, and the receptor, which is notch, its receptor name is Lin12, which is the notch receptor, and DSL is the signaling type. You can see DSL is giving signal to the nearby cell. The notch is receiving it, and that is why it is suppressing the primary fate. So you can see it is inhibiting the primary fate, which is generally activated in the central cell, and it is going for, that is why it is going for a secondary fate. DSL activates lin12 on neighboring cell fate, thereby promoting the secondary fate, which inhibits the LED23 signaling. Lin12 notch is activated by ligand in only two of the multipotential vulval precursor cell that will go for the secondary fate. Okay. And if lin12, if you topically activate in other VPCs, even if you activate in the nearby third VPCs, those will, instead of taking up tertiary fate, even those will take up the secondary fate. That is when the multivulva phenotype is achieved. Okay. Just wanted to tell you about the concept wheel book. If anyone wants to revise such kind of concepts, see, when it comes to Unit 5, there are a lot of concepts which are involved. And you will have to go through all these concepts, otherwise, you might be forgetting in between. Okay. So, this is a very good option. You can refer to it. That is the concept wheel book. All the points that you need to remember, CDSL, lin12, LED23, they will be all in one place, and they are all colored, so recollection during exam becomes very easy. All right, next question. Yeah, so next question is from sex development in Drosophila and humans also. The individuals considered in this question are having two haploid set of autosomes and no Y chromosome. So this is a very important hint to solve the question. No Y chromosome means what? If it are talking in case of humans, then it cannot be a male at all, isn't it? There is no Y chromosome means with A, one is not possible at all, because considered in this question, the individuals considered in this question. So if you look at the options, A1, if you have means that cannot be the answer. So here you have A1, right? Where else you have A1? You can see here also you have A1. So definitely 2 and 3 cannot be the answer, because they are considering Y chromosomes. I mean, they are saying there is no Y chromosome. And 2 haploid set of autosome. Okay, so that means that you are having um, the regular set only, and 2 means it's a regular cell, right? So X to A ratio, you might be knowing that in Drosophila, the sex ratio is done by the, means the sex development is by the X, so sex chromosomes divided by autosome ratio, because in them generally the number is not fixed. Okay, you might have different X and A ratio. So if the X by A ratio is 1, so you can have a look here. Okay, anyways, you saw the answer. So we have the, these are the X chromosomes and these are the autosomes, right? So these are the X chromosomes and autosomes. You can see either you can have 4 of each, or 3 of each, and 2 of each. In all these cases, the ratio will be 1, right? This will lead to the production of a normal female, all right? Then we have the meta female. So here the X chromosomes will be more and the Y autosomes will be less. So 4 by 3 or 3 by 2. In that case, you will have the ratio higher than 1. So either 1.33 or 1.5. That means there will be more female like characteristics. Okay. And in case the ratio is less than 1, for example, if it is 0.5, autosomes exactly double the amount of X chromosome, or more than that, or means exactly double means then it will form male characteristics. Okay, but if it is even less than 0.5, then it is meta male, and in case it is between 1 and 0.5, it will form intersex. So this way the sex determination mechanisms in mammals and Drosophila are very different. In Drosophila, it is a balance of this, and for humans, since it is mentioned that there are no Y chromosomes, hence the primary sex determination will be female only. Okay. Now, if the X is to A ratio is 0.5, it means only one X chromosome is there. You can see that zero point 0.5 means if it has to be male, then it is given also. See, when it is 0.5, it will be 1 and 2, right? Or it will be 2 or 4. Isn't it? Then only it will come to 0.5. That is X to A ratio. Correct? So that is why correct. So that is why if it is 1 by 2, but in that case also, even if it is 2 by 4 means it will be 0.5 only it is coming. It means only one X chromosome will be there, so that is why there will be no bar bodies. Only one X chromosome is there means why bar body will be required, isn't it? And let's say if the ratio is 2, so 2 means what? If the ratio has to be 2, X by A ratio is 2, X by A ratio. Okay, if it is 2 means what? Autosomes will be, there are 2 set of autosomes. So denominator has to be 2, because already they have mentioned 2 set of this is there, 2 set of autosomes. So in case it is 2, then if you have to maintain the ratio, then this has to be 4, isn't it? Then only it will come to 2 is to 1, right? So autosomes already they have said it is 2, X chromosome it will be 4. And if 4 are there, then out of that, 3 will be bar bodies. Only one X chromosome will be functional. Rest 3 will be bar bodies. So that is why if you are taking Drosophila and ratio is 2 means it is more than 1. So definitely it will be meta female. So 2 should be B, 3, and in that 3 will be bar bodies. 1 will be normal X and 3 will be bar bodies. Okay. So we'll have 2B3D. Got it? So these kind of combinations you have to make, and for humans it is pretty simple. 0.5 of human, it means that anyways they have said it is the female only, so obviously it will be female, right? And um, in the first case, if it is zero point 0.5, then you will have no bar bodies, because already it has said zero point 0.5. That means in humans, it means that there is no, only one X chromosome is there. Okay, already they have said no Y chromosome. That means zero bar body. But if it is two, and if we are considering humans, right? So then also it will lead to female only. There is nothing as meta female. But as I said, it will have three bar bodies. Okay. But in Drosophila, there is no bar body concept, because it is based on the ratio only. So that's why correct option is option 1. Okay, the reason why the X chromosomes, why it is X by A ratio. The reason is because, see, when the ratio is 1 is to 1, both the X chromosome and autosomes, they are equal number. That's why the ratio is 1. You can see the color is given equal. But when the it is autosomes are double the number of X chromosomes, you can see the color of the autosomes are more than the X chromosome. The reason is because there are some genes on this X chromosome and autosomes. Okay, and it's the genes that will control the sex determination. If you have more of autosomes, then these are not functional. These cannot have the effect. And these X chromosomes, these have a lot of effect in the female phenotype formation. How do they do that? They activate the expression of a particular molecule called as SXL. It's called as sex lethal. Okay. But if these autosomes are more, then they cannot activate these X genes. That is why here you don't get SXL. And because SXL is not there, a certain TRA protein also cannot be made. And if TRA protein is not there, then the molecule DSX or double sex, it goes for a male specific splicing. These work on alternate splicing. If you have some molecules, required molecules, then it will go for male specific splicing, which will give the male phenotype. But if the SXL is present, then it will activate the TRA and TRA2, and in that case, it will activate the expression of DSX in a female specific manner. In that case, you will get the females. That is why when the ratio is 1 is to 1, you get female. And if you get 1 is to 2, then in that case, you get male. Okay, where the ratio is 0.5. So next question is also about Drosophila, but I'll skip this. Let's see some other type of questions. You can try this on your own later, right? Now this is regarding Dictyostelium discoideum. Even this is a popular question in CSIR. Every year, every 2 exam papers, you might see a question, 2 or 3. Now Dictyostelium discoideum is a very interesting organism. It can exist in singularity also, single cellular form, and multicellular form also it can exist. Okay. So how can that be possible? Generally, you know that if you're talking of a normal embryonic development, a multicellular organism will come from a single cell by lot of cleavage division. But here, how does this organism get multicellular? It gets by aggregation. Single cells will send off some CAM signaling, because of which the other cells come close and they will form an aggregate. So this CAM signaling plays a very important role in the development. And once this aggregate has formed, this can even initiate, it will behave like an organism. It will have an anterior direction, a posterior direction. It can migrate also like a slug. It can migrate on the surface. It can even form a standing slug, and later from here the fruiting body can develop, spores can form, and when the spores will hatch out, again organisms will be released. Those will be again living as single cell lifestyle. So this is a very weird organism. However, this morphogen, that is CAM signaling, is synthesized by different adenylyl cyclases. Why it is called morphogen? Because it is affecting the morphology of this organism. They are expressed at different stages of its life cycle. The following statements, that is the P2S, they refer to the effect of mutations in different adenylyl cyclase genes. Okay, so which out of this is correct? One is ACA. The different adenylyl cyclase, you have ACA, ACB, and ACG. All of them have different roles. So which out of this is correct? Anyone knows what will be the answer to this? Or shall I explain? Any guesses? Has anyone studied this part? Okay, you're saying option 4, Sarita? All right. So let's have a look at the growth phase and the differentiation phase. See, as I told you, these individual cells, they are aggregating together. See, aggregate competent cells by CAM signaling, they have aggregated. They have formed a mound and a tipped aggregate. So here, different signaling goes on between the two parts of cells, okay, two categories of cells. And they form fruiting body spores, which again leads to single cellularity, right? Now, between the pre-spore cell and the pre-stalk cell, in this entire slug, there is a categorization. The first part, or the one-fifth part, you can say, one-fifth part is the pre-stalk cell, and the fifth part is what is the pre-spore cell. And there is a signaling between them as well. Okay, you can see how they are signaling each other. So, option 1 is the right answer, because addition, if you add exogenous CAM pulses to ACA minus cell, ACA minus means they cannot, first of all, you should know what this ACA, ACB are all involved in. ACA is involved in the initial distribution of the messages, where the single cells will be sending the other cells, right? So one layer of cells, after getting the after getting the signal, it will come closer. Okay. Now this set of cells starts releasing the signal. Then once this also will come closer, so that way waves of camp go on, and to produce this exogenous camp outside the cell, ACA is necessary, right? So addition of exogenous camp pulses to ACA minus cell, even if if the cell is ACA minus, which means it cannot release the regular camp, but if you add camp from the outside source, it will restore the normal gene expression and development, which means that P is correct. So P is talking about the ACA deficient cells, they can be allowed to aggregate by exposing them to pulses of camp. Okay. Now ACB deficient cells would form normal fruiting bodies, and the spores germinate when exposed to favorable conditions. So what is ACB involved in? ACB mainly is responsible for the increase in adenylyl cyclase activity that will happen after aggregation. So that way you can remember, after ACA it is ACB, and with this ACB minus cells, they will show normal development until the slug stage. Till the slug stage, it is the culmination and aggregation part. So this will happen normally. Okay. After which the ACB part is to form the fruiting bodies and all. So if it is not present, if it is ACB minus, then it will lead to abnormal fruiting body. It is not possible that with ACB minus you will have normal fruiting bodies. In fact, the spores also that will be formed, they will be abnormal. That is why Q is incorrect, because it is saying that it forms normal fruiting bodies. ACG deficient cells develop normally, and the spores germinate in the spore head itself. Now, what is the function of ACG? ACG is exclusively expressed after the fruiting body has formed. So, see, all of these are in sequence. First ACA, then ACB, and then this ACG. Their function comes in sequence. Okay. So, now the fruiting body has formed, it has to germinate. Germinate means it has to hatch out. Hatch out means the proper word is germination, so the single cells can be released. And this is done by ACG. It is controlled by ACG. Okay. Cells lacking ACG develop into normal fruiting body, because anyways fruiting body was a function of ACB. So even if the cells don't have ACG, still fruiting bodies will form. They will also have viable spores also, but the germination part won't happen properly. Okay, because ACG is not there to control now, so they won't know when to germinate, right? So that's why R is also wrong, Q is also wrong. ACG deficient cells, they will develop normally, but the spores will not germinate. Okay, they will not germinate. Next, spores from the ACG deficient cells will germinate irrespective of the osmotic condition. So no matter how much is germination, how much is the osmotic condition, actually ACG helps to germinate with osmotic conditions. Okay. See, in wild type cells, heat shock stimulated germination occurs synchronously at low osmolarity, and it will be inhibited in the presence of high concentration of sucrose, 250 millimolar concentration. But in contrast, if you take ACG minus cells, they will germinate spontaneously even in high osmolarity. That means what? If the sucrose concentration, if the sugar concentration is more, then these cells will not germinate. ACG will not allow the cells to germinate in high sugar concentration. They will germinate only in the low osmolarity effect. Okay, but if ACG minus cells are there, they cannot control. So even in high osmolarity also they will germinate. No matter whatever the conditions, ACG minus cells will germinate irrespective of the outer conditions. Okay, so that is what actually is abnormal germination. They will display abnormalities in the germination process. Okay, so hence S is correct. Dormant spores, they have high camp levels that decrease just before germination, and it has been established that high spore osmolarity maintained by the presence of 100 millimolar ammonium phosphate maintains high level of camp and inhibits the germination. During germination, ACG therefore acts as an osmosensor. In conditions of high osmolarity, activation of this ACG gives rise to high level of camp that ultimately lead to activation of PK and to the maintenance of dormancy. So if the cells have a constitutive PK activity, then they germinate very poorly. For proper germination to take place, camp levels should be decreased. But if they are not decreased, then the germination process is not proper. Okay. Then the last question is regarding this limb development. Even this is a very important concept from Unit C, Organogenesis, Unit 5C. In mice, the gene encoding TBX5. So generally, this TBX4 and TBX5, these are involved in the forelimb and the hind limb formation respectively. TBX is responsible for making the specification of forelimbs. It is transcribed in the limb fields of the forelimb, while the genes encoding islet1, TBX4 and PITX1, they are expressed in the presumptive hind limbs. Okay, so following statements are made about limb development in mouse. That is, loss of TBX5 gene leads in complete failure of forelimb formation. So obviously this has to be corrected because already they have said that TBX5 is in limb field of the forelimb. So if TBX5 is not there, then forelimb specification will not happen at all. Okay. Hind limb bud growth and initial patterning appears normal when TBX4 is knocked out, although leg development is arrested prematurely. So, how is this possible? TBX4 controls the hind limb, isn't it? So, if the hind limb is knocked out, then how can you expect that the hind limb bud growth means initial patterning might happen, but the leg growth obviously you won't expect it to be formed. Okay. Now, misexpression of PTEX1 in forelimb ceases development of muscles, bones, and tendons. Now, have a look at how this works. This PTEX1, PTEX1, it is expressed in the hind limbs, but not in the forelimbs. Okay, and you can see that TBX5 expression you can see only in the forelimb part, but not in the hind limb part, right? So, hind limb bud growth appears normal when TBX. So, leg development will be arrested, but misexpression of PTEX1 in the mouse, it will cause the muscles and bones and tendons, it will cause to develop, but here it is saying that it will cease the development. Okay, and when the islet one is inactivated specifically in the lateral plate mesodome, the hind limb still develop. You can see that islet is expressed in the hind limb. Okay, so when islet is inactivated, hind limbs do not form. Obviously islet is responsible for hind limb formation, so that is why, since it is saying activated, inactivated specifically, still hind limbs will develop. This is wrong. So option C and option D is wrong. So that's why the correct answer is option 1. Okay. So that's all from my end. There is one more question regarding the limb development, and this is with respect to the SHH signaling. You people can try solving it at home and let me know in the comment section what is the answer. Okay. So that's all from my end, and that's all from Unit 5. Your Unit 5 sessions are done for this series. All right. All the best for your exam, and let us know in case you're clearing Biotecnika. You're a Biotecnika student. Do let us know. Thank you.